Hi, welcome back. In the previous session, we were looking at proteins, and what we have here is a video of how hemoglobin functions. We will go it through very fast before we embark onto the new unit that we are on. So, hemoglobin is made of globin proteins and an iron-rich compound known as heme. It is found in red blood cells and is involved in the transportation of oxygen. Red blood cells do not have a nucleus, so increasing the amount of space for hemoglobin. There are approximately 3 million hemoglobin molecules in each red blood cell. So if we have one red blood cell, the structure of hemoglobin enables it to carry oxygen with high efficiency. So it has a quaternary structure and is made of 4 globin units, most commonly known as, we can't go into these details, but you can see the structure that I wanted us to see here, that normally the alpha chain of the protein will have these sites. This is iron with a site. So when this one rotates, of course, as an ion atom and a heme group, these have association. This is oxygen molecule. When oxygen molecules attaches to one, it allows it to rotate so that it will attach to another one and another one. So this structure enables hemoglobin to be able to carry oxygen efficiently. So you can see what happens here. When this one attaches, it opens a way for other oxygens to attach on the molecule and so forth. So this conformation in protein makes it easy for oxygen to bind to heme groups. This makes it easy for it to bind in heme groups, and this is very essential. As we said, normally efficiency depends on structure. Efficiency depends on structure, just like oxygen here also, just like how hemoglobin does this. So we will embark on our unit for today. We will embark on the unit for today, which is about nucleic acids. So with nucleic acids, the detailed instructions to build proteins are stored in extremely long carbon-based molecules. So as we look at nucleic acids, they are very essential. Why are they very essential? It's because even proteins themselves depend on nucleic acid. So we say, so instructions to build proteins are stored in nucleic acids. That is one important factor. So instructions are to build proteins are stored in extremely long carbon-based molecules called nucleic acids. acids. Nucleic acids are polymers made of, they are polymers made of monomers, just like made of monomers called nucleotides. So unlike in proteins where we had amino acids, here they are made of nucleotides. So these monomers the monomers are called nucleotides. So nucleotides are composed of sugar. So the structure of nucleotide is very important. It is composed of sugar. It's composed of sugar, a phosphate group, a phosphate group, a phosphate group, a phosphate group, and a nitrogen containing molecule, and a nitrogen containing molecule. And a nitrogen containing molecule called a base. Called a base. So that is as far as the structure of a nucleic acid is concerned. It has three parts. That is the sugar, a phosphate group, and a nitrogen containing molecule or a base. So there are two types of nucleic acids. Types of nucleic acids, and these are the DNA and the RNA. And both the two are very essential in terms of production of proteins. So nucleic acids work together to make proteins. Nucleic acids work together. They work together to make proteins. They work together to make proteins. So what happens here is DNA stores the information for putting amino acids together. DNA stores information for putting amino acids together. So for putting amino acids together to make proteins. So it is responsible for storing this essential information that is responsible for making different amino acids which will make proteins. And RNA helps to build proteins. RNA helps to build proteins. RNA helps to build proteins. DNA is the basis of genes and heredity. DNA is the basis of genes and heredity. DNA is the basis of genes and heredity. So the basis of genes and heredity, but cannot do anything by itself. But cannot do anything by itself. So it is essential for us to know that it cannot do anything by itself unless, of course, the orders are placed in the right structure. So instead, the structure of the DNA, which is the order of the nucleotide, provides the codes for the proper assemblage of proteins. So it provides the structure of DNA, I mean the order of nucleotides. Provides the code. Provides the code for the proper assembly of proteins. Proper assembly of proteins. So many kinds of RNA molecules assist in assembling proteins based on the DNA code. So RNA can also catalyze reactions. So RNA assists in assemblage. Assist in assemblage and also in assembling the right codes, in assembling proteins based on the DNA. Proteins based on the DNA. RNA may even catalyze reactions. RNA may even catalyze reactions. We will be looking at this short video to try and see how it. So what we have here is a model of the DNA, which of course is made of nucleotides. DNA and RNA are nucleic acids, polymers made of subunits. This is a close-up of DNA structure. So we are saying the DNA and RNA are nucleic acid polymers made of subunits called nucleotides. So these are nucleotides. So here you can see we also said that this is the DNA nucleotides. As we can see them, we said the DNA nucleotide is made of 3 parts. It's made of the 3 parts. You can think about them. The 3 parts included sugar, then it also included a sugar, it included a base, and also this same structure included the last part of the 3 parts, which of course is nothing but nitrogenous base. So if we look carefully here, this is the structure. This one is the phosphate group, and this is the nucleotide, and this is the sugar, and we are having the nitrogen base. So we are having the nitrogen base. So the sugar can be ribose or deoxyribose. This is a deoxyribose in DNA and ribose in RNA. We can also have the phosphate group. We can have, as you arrange them, you can tell that you can choose the correct one for DNA and RNA depending on the sugar. So the RNA will have deoxyribose sugar, the DNA, while the RNA will have the ribose sugar. That is the difference. And the nitrogen base stands and remains the same. So the DNA and RNA are each composed of 4 different nucleotides which differ in the nitrogenous bases. 3 of the 4 bases are the same in the DNA and RNA. We are not going into these details. We will be seeing them in the next unit. For now, we will stop here, and in the next lesson, we will be looking at something else.